Okay, very good afternoon to everybody. My name is Dr. Ahmad Khaldun Ismail. I'm the emergency physician from the National University Hospital in Kuala Lumpur, Malaysia. Would like to welcome everyone to our evening webinar on a Sunday, um, uh, 21st March 2021. We have a very interesting topic to discuss this evening. The title of our webinar is Snake Bites in Pets. So this is a continuation of the first webinar we had late last year regarding snake bite in pets. That was an introductory lecture about snake bites and how to manage snake bites in pets. So for this year, we have another lineup of experts to discuss and to share some experience regarding snake bite and also various snake species that we have in this region. So this webinar is organized by the Forest Research Institute of Malaysia, FRIM, the Malaysian Society on Toxinology, MST, and also supported by the Malaysian Society of Veterinary Medicine, MAVMA. And this program is also providing the CPD points for doctors and veterinarians. So without further delay, I would like to first introduce our program for today. Our agenda is first to have a bit of announcement by our moderators regarding our housekeeping, and then they will be introducing our speakers one by one. And we will have 4 speakers. And before we proceed with our forum towards the end, and that is where we have the Q&A session. For the Q&A session, I would like to invite everybody to post your questions in the Q&A box, not in the chat box. Okay, please submit your question in the Q&A box that is in your Zoom, and we will direct, the moderators will direct the questions to the speakers during the forum. So again, I would like to inform that this program is providing CPD points for doctors and also CPD points for veterinarians. Towards the end, we will provide the QR code for the CPD points for doctors in Malaysia. So you can scan using your MMA QR code scanner towards the end of the program. And we will also have a feedback form. And in the feedback form, after submitting the feedback form via the link provided later, all participants will be receiving their e-certificate as well. Okay, so that is very important because this is auto-generated e-certificate. So you need to fill up the form using the link provided later on. Okay, so without further ado, I would like to introduce our 2 moderators for today. First is Norsham Yaakob. She's a research officer and the head of the Eco-Tourism section from the Forest Research Institute of Malaysia. And our second moderator is Ho Wai Mun. She's also a research officer in the Ecophysiology branch of the Forest Research Institute of Malaysia. So without further delay, I would like to introduce I would like to pass the mic to our moderator, Norsham and Ho Wai Mun.
Thank you, Prof. Khaldun. My name is Norsham, and I will be one of the moderators today. With me here is, I'm going to share. Okay, this is not a product placement for the G-Shock Casio watch. Maybe some of you guys who are divers may heard of this expensive frogman series. In 2011, Prof. Das and his team rediscovered the Borneo rainbow toad, a species thought to have been extinct since 1924. They found this reclusive toad in Gunung Perinsen, and then the discovery inspired the designers at the G-Shock to create the Frogman Borneo Rainbow Toad limited edition. So, which I think is a good way to celebrate this rare toad. So here I am sharing this with everyone because I am super proud and also super envious. Let me introduce Prof. Das. Yeah, he was born in India in 1964. He's still a young man. He's obtained his doctoral degree in animal ecology from the University of Oxford and was a Fulbright Fellow at the Museum of Comparative Zoology, Harvard University. And since 1998, he has been the staff of the UNIMAS and currently professor at the Institute of Biodiversity and Environmental Conservation. His current research interests includes herpetology, systematic biogeography, conservation biology, and history of natural forests, natural history. So without further ado, I would like to call upon Prof. Das.
And your wish is granted, Nosham. Thank you. Thank you for the generous introduction. And I'd like to thank the organizers for this opportunity to say a few words about a topic all of us are learning about. There's not much literature to review, so we are pretty much at the base of a steep hill. And all learning, maybe today a big learning session for all of us resource persons. I will talk about the snakes of potential interest to people with pets, dogs and cats in this country, and could be some other animals elsewhere. And as for the G-Shock, there's also one on the python issued last year. So those interested in snakes can maybe have a look at the python G-Shock. So I'm going to, I know I have only 20 minutes, so I'll try to share screen and start. All right, can you see my screen? Someone can tell me?
Yes, yes.
Yes.
Thanks so much. So I'll talk about snakes of potential interest in this little field, vis-a-vis association with bites on pets of humans. And let's see, moves. Oops. All right. Okay, so I classified the talk into such a general introduction and how snakes can be a threat to pets, swallowers, the spitters, and the biters. So these are the three classifications. I hope they become standard one day and we can be held responsible for this classification. And then I'll make some very brief comments. I'm sure there'll be many more inputs into this. I would like to give my background as a zoologist with rather little knowledge of veterinary aspects. So I'm here to learn from all of you. But I do know a little bit about snakes, and I will share some of that with you. Although this webinar is supposed to be mostly on venomous snakes and their effect of the bites on pets, other snakes can be of importance, such as pythons, and there are several species in this country. The most important perhaps is the reticulated python, which in the old literature has been called Python reticulatus, or even Brog Hammerus reticulatus, but apparently the current name we should follow is also unwieldy Malayo python, which even city dwellers may not be aware. It exists in our midst and may explain the occasional disappearance of small dogs and cats. Even here in the city of Kuching and even Singapore and Kuala Lumpur, They are known to be present as Bomba sometimes removes them, fire department removes them from housing areas, and sometimes they're found to ascend, climb high rise, mostly through pipes. And of course, there are several cases of their bites of humans. And here on Borneo, there's a smaller python, but of course, unless it's a village near a forest, these are strictly forest dwelling species, you would not encounter them, but they also feed on small mammals and maybe birds. This is the Bornean short-tailed python, Python breitensteini. Whereas in Peninsula, you have another smaller python found in the forests, ecologically similar to the Bornean one, but more in fairly recent times, they've been shown to be two distinct species. This is a subadult Python Bronchosma I. So I'm sorry, the scientific names of most species are complex, and when it ends with I, most likely it is honoring a person. In this case, it's a Dutch herpetologist, Bronchosma, whereas this python is named after explorer from Austria, Breitenstein. So like it or not, we have to remember these scientific names because the common names are even less standardized. So different people call them by different names, but all of them have the same scientific name at any point of time. So this also is a predator of small mammals and maybe a predator of cats and small dogs if there are in the vicinity of villages. So, and up north and more common in Thailand and Peninsula is this very common species in the pet trade, but marginally present in Peninsular Malaysia, not in Borneo, is the Burmese rock python, which of course is a formidable predator and even known to attack humans. And I'm sure it would find it very easy to consume mid or large sized dogs and cats. And there are many papers if you search of and blogs of bites of on humans, and I found this one rather interesting, a case of an injury more serious than a laceration of a hand of a person. So this is maybe something we can take into consideration when dealing with bites of pythons on pets. So we now move to the more major, okay, one more species, not a python, not a venomous snake per se, but a species, a group of species called cat snakes. They can be quite large, more than 2 meters, and known to consume animals as big as mouse deer. So potential predator of cats and small dogs. The mangrove cat snake in the past, they were called Dendrophila, Boiga dendrophila, but right now there are different opinions on what the name for the names, I would say, for peninsular and Bornean species should be, Annectens or Melanota perhaps. So the jury is still out there. And again, species from mangroves, but given that there are dense habitation around mangroves, this could also be associated with the disappearance of cats and maybe small dogs. But although the venom of the species apparently, Dr. Scott will tell us more perhaps, is specific to birds. So I mentioned different ecological categories. Those were one. There are also snakes that not just have venom for bites, venom transmission through bites, but also through spitting, specifically in the eyes of animals, enemies or prey, perhaps. And certainly the Bornean population of the Sumatran cobra, again, there could be more than one species in this group of snakes, are capable of spitting venom, not as well developed as the African spitters, but many dogs get it in their eyes, and maybe cats as well, or when they try to attack, perhaps. This snake, unlike most other snakes, shows color change in uh, ontogeny, which means a juvenile has these very distinct bands, pale bands on a dark body, whereas adults lose these bands. So we don't know the function, maybe perhaps some ecological processes involved, maybe a mimicry of some other animal. Certainly the little ones look very similar to baby king cobras of Ophiophagus. So spitters are not well understood, and there are not much literature either. For the Asian species, there's a lot of literature on the African species. And here's one, interestingly published in the Medical Journal of the Armed Forces of India on snake venom ophthalmia. And of course, the species would be different in these different parts of the world. I'm not sure, they didn't recognize or identify what species it was there. But here we know the Javan one, of course, not found in Malaysia, but in this country, only the Bornean population confirmed, as far as I know, to spit venom. And finally, we move to the more, the major snakes of medical importance, which are those that conduct venom through biting. And these include a variety of different ecological and morphological types. There are front fang ones like cobras and kraits and vipers, and there are also back fang ones, which are also venomous, but their venom teeth, the fangs, are located midway along the maxilla, the head, the oral cavity. And so they need to have a really secure grip to bite, and yet they're implicated with many bites in this country. Many are not reported. Some people recover, and some have no envenomation, but these are really poorly studied group of snakes, back-fanged. There is a book on this by Dr. Weinstein, which I'm sure we will use in the future if people get active in this field of dealing with pets and snake bites. There are also different ecological types of snakes, and this is very important to understand what snakes bite pets: sedentary, active foragers. Especially the active foragers are more of a threat. These include cobras. Kraits are also active foragers, but they are specialists of other snakes. So there are probably fewer bites for kraits than by cobras. So pretty much most of the bites, I would assume, are by cobras of some species. So talking about the species not really important for pets, but certainly very, very important for humans is the krait, the common Malayan krait. Very strange distribution. This is the unbanded morph from Bali, but of course we know the populations from Peninsula, Sumatra, and Java have bands. Strangely, the species doesn't occur in Borneo, but maybe it does, because I've seen, someone has shown me a picture of one in a remote area where it's unlikely to be introduced. So maybe it's present in certain restricted areas. So very, very severe envenomation possible, and neurotoxin associated with its bite. And of course, that Thai Red Cross is a source of the antivenom serum. And another very familiar crate is the bandit crate. And again, a complex of species. These are not that many cases associated with its bite. I have two images. I've taken the top one in Brunei, in Borneo, and the bottom one in Bhutan. And of course, the scales and the color, the patterns are quite distinct. And maybe, yes, more work needs to be done to understand its identity and taxonomy. And a photo of a red-headed crate. I think I'm going to go a bit fast. I'm maybe close to my allocated time. And the blue coral snake, again, a possible species of medical importance to humans and pets. But there has been, strangely, even for this common species, little information on its bite and treatment. And its relative, a very, very small species, Caliophis intestinalis, small, but possibly of great medical importance. It's hardly 15 centimeters in length, 12 centimeters to 15, but it has a venom gland about a third the length of his body. So certainly not to be taken seriously, lightly. And there have been a couple of bites, I know, but of course ended happily for the patient, human patient. So for Peninsular Malaysia, the major species associated with bites would be the Sumatran Cobra and the monocle cobra, Naja cautia. These are highly adapted species found in forests and edges, plantations, and of course, very, very serious bites and deaths recorded for both humans and their pets. This animal is from northeastern India. And as I mentioned, Sumatran cobra, juvenile and adult. And of course, the largest of the snakes, but fortunately for humans, it's Ophiophagus. It feeds on other species of snakes and sometimes monitors. And in the wild would not touch mammals. But nonetheless, many people keep them as pets or use them for shows or in zoos. So the potential for conflict between the king cobra and mammals like humans and our livestock remain. Again, I say this is a species complex. There are several species in this group, maybe 4 or 5. We are actually working on this problem, and of course, extremely dangerous snake. I mentioned active foragers perhaps present a greater threat to pets than sedentary sit and wait kind of predators. Like the Malayan pit viper is that category, would stay in a certain place. And I'm not sure if there are any records. Perhaps the panel here or the participants can to tell us all, enlighten us on the likelihood of bites by this species on pets. Certainly if a dog or a cat discovers a quiet snake in the undergrowth, may try to bite it and therefore get bitten. So these then would be a very, very serious case. So if you look at the literature, not much published on bites by snakes and dogs for Asia. There is a lot for other parts of the world like Australia or Africa or North America. So big scope for, huge scope for research by people who are interested in this field. And certainly very important to document all observations and further research. So my last bit would be on how we can go about trying to identify them. Of course, the first, the priority is to recognize a venomous snake from a non-venomous snake, and later others can discuss how to figure out whether there has been any envenomation or not. And of course, in the past, people were told, okay, check the neck of the snake. If it's narrowed at the neck, it's a viper. If it's not, it's something else. But of course, we know it's much more complex than that. And the resources we have are several. Some of us, after a while, get quite good in looking at the morphology of individual species and can therefore identify species, but many of them are so similar that visual identifications are difficult sometimes, especially in areas like here, where there are so many species and many are undescribed. So taxonomists, as some of us call, use a device called a taxonomic key, which includes steps like this to recognize them. But this involves knowing these character systems, such as the head of a snake has a lot of names, and it takes almost a lifetime to know them. So, but yet this is the only foolproof method for accurate identification of species. And for rapid identification, at least the major, it's important to know the major scale counts of snakes, the mid-body scale rows, ventrals, and subcaudals. So a mid-body is counting the scales from the ventrals, the broad belly scales, obliquely up to the middle of the body, and multiplying that by 2. Plus you have azygous, the central one. So it's almost always an odd number. So that's the mid-body scale count. And you can also have the ventral scale count. This starts from the broad scales on the belly, but excluding about 3 or 4 small scales, which don't touch the the scales on the flanks. So up to the vent of the snake. So this is the ventral scale count. And then it's very also important to count the scales on the underside of the tail, the subcaudals. And of course, there's a difference between male and female subcaudals. The males have longer tail and therefore more scales. So these are essential for accurate identification of snake. But you can also photograph a snake and send it to someone who knows snakes with a mention where it was taken for a almost certain identification. And if the snake is dead, of course, it's probably illegal in most parts of the world to kill, harm biodiversity, even if a snake's involved in a bite. But if it's dead, maybe preserve it for identification later, or kept safely in a bottle and released subsequently. So these are ways to recognize snakes. Books, read as much as we can, field guides and journals, which have the most recent research, identification charts. These are harder to update. Websites are good sources, and people like Professor Khaldun has different ways of getting in touch with him with images of snakes for identification, and then recommendation for the treatment. So final word, I thought more about this. I think pets are very naive prey, but they're naturalized. They don't belong to an area, and therefore they're not aware of the predators. As the predators are native and the prey are naive. And I found this interesting contrast to island endemics, where the naive prey are predated by introduced predators. So in the end, in both cases, it's a disaster. So therefore, I end my presentation by saying pets need special protection and security. They don't know the world that they live in. Thank you. And this is the last image I have. This was taken about 20 years ago. Nosham was present, and a very famous American herpetologist hurt himself severely at the excitement of this discovery. He crashed through the board of, I think, Sungai Buloh, and hurt himself pretty bad. But of course, the snake was bagged, and we took this picture later. Thank you so much.
Thank you, Professor Das. Just a quick reminder for everyone, if you have any question, please type it in the Q&A box. The speakers may reply directly to you, or the moderators will help to raise the questions later if your questions have not been answered yet. Coming up is our next speaker, Dr. Chu Hawk, who is currently a senior lecturer at the Venom Research and Toxicology Laboratory of the Department of Pharmacology, Faculty of Medicine in University Malaya. His areas of expertise are medical pharmacology and toxinology, with a special interest in snake venoms and toxins, as well as anti-venom research. He has also published more than 60 peer-reviewed articles. Can we have the slide on Dr. Tan, please? I'll just continue with his CV. Dr. Tan is a registered medical practitioner and also a life member of International Society on Toxinology. He serves as the Honorary Secretary for the Malaysian Society on Toxinology, a faculty member of the ASEAN Marine and Snake Envenomation Management Symposium, or AMSAM, as well as the Toxinological Society of India conferences. He has also received the International Award of Young Scientist from the Japanese Association of Laboratory Animal Sciences. Let's invite Dr. Tan for his talk on snake venom composition, toxicity, and neutralization.
Thank you very much. Thank you, Waiman, for the very kind introduction. Just to check, can you all hear me?
Yes.
And the slide is on? Okay, good. All right. So thank you for the invitation. I'll keep my talk to 20 minutes. As has been introduced, I'm actually not a veterinarian. I'm more of a medical scientist. So my presentation might be slightly biased towards the venom study. Nevertheless, of course, I think envenomation in pets is equally a problem as well. So I hope that, at the end, we can get some feedback from our veterinarian friends as well. Yeah. So enlighten me on the veterinarian aspect of snake venom. Okay. The first of all, I'll perhaps just give a very brief introduction about these poisons and venom. So a lot of time people get confused between the two words, poison and venom. But of course, just to get things right, strictly speaking, they are both different things. Yeah. So in short, if something is poisonous, you bite it, you die. And if something is venomous, it bites you, then you die. So that's just as simple as that. The definition can be more complete in the sense that you can say that poisons, it can be something or produced by organism, or it could be equally a synthetic thing. So usually poisoning occurs through ingestion, inhalation, absorption, through skin and so on. So it's a more passive kind of way. Whereas venom is usually a secretion produced by specialized tissue or glands in animals, then it has got to be delivered to animals, yeah, or to recipients by inflicting a wound, no matter how small the wound could be. So it is more like an active kind of delivery compared to poisons. Yeah? So these are all the venomous snakes. I mean, sorry, not the snakes, but animals. In fact, some of these are pets to some people, yeah, okay, so they can be pets as well. Now, so snake venom, it is produced as I mentioned just now, primarily in the venom gland, and then it has got to be delivered through a specialized system, in which case for snake, it will be the venomous, the venom fang, yeah? Okay, so this is where the venom will be channeled in and then given through the, injected into the tissues of the recipient through the fangs. And if you look at, between the Viperidae and also the Elapidae snakes, Elapids will be snakes like cobras and so on. And then Viperids will be pit vipers and vipers. Both are the major groups of venomous snakes anyway. So their fangs are slightly different. Yeah, one is small, fits, and usually quite small. The other one is more flexible. Yeah, this is the viper or viperid fangs. Okay, more elongated. Okay, now in terms of the venom composition, if you look into the venom itself, yeah, so this is a venom. So of course, in laboratory, we have various ways to characterize the composition. So for usually it's biochemical means like HPLC, SDSPH, LCMS, and so on. In brief, it is a highly complex mixture of various bioactive compounds. These compounds could be, consist of various forms of proteins, peptides, even nucleotides, some salts, ions, and so on. But the majority of the composition is proteins. So between the elapids and viperids, these are the two main groups of snakes, venomous snakes. So to cut things short, but of course it's not always the case. There's always exception. But in brief, if you look into the venoms, the elapids like cobras, for example, cricks, for example, the venoms usually are more, dominated by low molecular weight proteins, for example, like the three-finger toxins, phospholipases A2. Whereas for the viperid venoms, usually they are more complex in the sense that they contain a lot more high molecular weight proteins. Yeah? Okay. So are they different? Of course. Each of these toxin components, they have different pharmacological activities. And together in the mixture, in the cocktail, they access the activities, which could be sort of like synergistic or complementary to each other in inducing the venom toxicity. So we will look at the elapid snakes like cobras, cricks, and sea snakes. You will see that, again, that their venom contains abundant proteins of low molecular weight, in particular neurotoxins and cytotoxins. Yeah. But I must emphasize that the toxin types and abundances, means how much each toxin is in a venom, vary across species. Different species, even though they are the same cobra, people call it cobra, but there are many species. Now, the toxin subtypes and the diversity can be quite different. And of course, more research is also showing, even within the same species, sometimes you will also see variation in the snake venom composition. So this is a very brief introduction, just to let you know that cobras, this is perhaps one of the most worrying for us and also for our pets, because you see them at home, so sometimes they get into our home as well. So cobras, their venom is predominated by alpha neurotoxins, and then cytotoxins and phospholipase A2. For cricks, it is also some alpha neurotoxins, but their main venom toxins will be the beta bungarotoxins, and then you have other enzymes and other components as well. For sea snake, primarily the venom contains alpha neurotoxins and also some enzymes like phospholipases A2. Now, I'll just give a brief introduction about the neurotoxicity that is caused by elapid snakes. For example, like cobras, cricks, king cobra, and also sea snakes in Southeast Asia, of course. So when you talk about alpha neurotoxins, Okay, so this is a diagram showing the nerve, you know, the motor nerve, and then this is the muscle. So you have this neuromuscular junction. Yeah. Okay. So under normal circumstances, the muscle can contract because of the acetylcholine as a neurotransmitter. Yeah, that actually stimulates the contraction of the muscle. Now, in the presence of alpha neurotoxin, it blocks the nicotinic receptor, and then it disrupts the neurotransmission. In the case of beta bungarotoxin, the beta bungarotoxins damages the nerve terminal presynaptically. Yeah, okay. So both alpha and beta neurotoxins, in short, they cause neurotoxicity. Yeah? Okay, and that effect in clinical, of course, will be paralysis and then respiratory failure. The same will happen to pets as well. Now, this is an example of a Najakalphia, a cobra in Southeast Asia. Yeah, so you have this in, of course, Thailand, Vietnam, Malaysia, yeah, and some other places as well. But the research found that actually the venoms, even though belongs to the same species, but they can actually be quite varying in terms of the composition, where the Thailand one contains a lot more neurotoxins, yeah, and this translated to pharmacologically, yeah, you will see that the venom actually has different neurotoxic effect, and also the LD50 or the lethal doses also different accordingly. Now, in terms of viperine snakes, okay, the toxins it contains more complicated, I would say, yeah, okay, more high molecular weight proteins or high molecular weight enzymes. And a lot of these toxins, I know this is a bit generalized, but they actually cause bleeding as the final toxicity. These various hemotoxins from viperics, they can act at multiple pathways or multiple levels. So your clotting cascade, it can either use up the clotting factors, or it inhibit the certain clotting factors, or it can be even inhibits the platelet plug formation, yeah, or sometimes break down the fibrin clot. Yeah, so eventually the patient or the victim will succumb to bleeding disorder. Okay, all right. Now, towards the last part of my talk, that's about anti-venom neutralization. Okay, so treatment is a lot. Okay, maybe you're better than me also in terms of how to treat a pet envenomed by snake. But I know that this is also being used. That is the anti-venom. Yeah, so anti-venom is of course developed usually for humans, but I think in some cases also they use it for animals as well, I mean for pets. So in short, what is anti-venom? Is basically treatment, biological treatments that contains antibodies or derivatives produced from antibody that can be used to neutralize snake venom effects, hence the treatment. So how anti-venom is produced, I think this is quite straightforward. Typically, the anti-venom is produced through hyperimmunization in big animals like horses. Yeah, so you get the anti-serum, you purify it, then you get the antibody from there on. Yeah, okay, and this is then packaged and distributed to various locales. The production and the use of anti-venom, however, can be quite there's a lot of debate about it because unlike most of the medicines for use, antivenom is actually quite species specific, which means if an antivenom is produced to treat envenomation by a certain pit vipers, it may not be effective if you use it to treat envenomation by another vipers. Yeah, okay. So sometimes this is what we call species specificity of the antivenom. Yeah, and as I mentioned just now also, sometimes even for the same species, yeah, you will see also variation in the antivenom efficacy. And this is largely because of the snake venom variability, yeah, differences in the snake venom composition. Reason behind it, there are many. Could be because of geographical factors, could be because of ontogenic factors in the snake itself. Yeah. Okay. For example, like a Naja Kaufia, the monocle cobra in Malaysia, is slightly different from that in Thailand. Yeah, because the Thailand one contains a lot more neurotoxins. Okay. So this is an example of it. Because of the venom composition differences, so that also brings along the differences in their antigenicity. Yeah, okay. So the problem will arise when an anti-venom is produced by a certain country against the population, a certain population of the snakes in that area. Yeah, okay. So the questions here is, many people ask this question, whether there is one anti-venom that can be used for all species. The answer is not likely, is no, yeah, okay. And then the second question that follows that will be, so which anti-venom should I use? Of course, this has to be very species and also locality specific. It will be best to consult the authority, yeah, okay, in the area on what anti-venom that you can use to treat your pet. A lot of time is many times it actually rely on the species that is causing the envenomation. Okay. So from the laboratory aspects, of course, we do have some evidence to show that antivenoms against different species, sometimes you can depend on the cross neutralization effect. Yeah. So for example, like this is a monocle cobra antivenom produced in Thailand, but you may also use it to treat envenomation by other species, for example, like Naja sumatrana or even Naja sputertrix as well. Yeah. Okay. Because of the cross neutralization effect. Yeah. Okay. So for example, like this is another study, the Philippines antivenom that is for Naja philippinensis, but it is also seems to be effective for the other species in the area, which is Namasa samarensis. But the doses may be different. Yeah. Okay. It probably because it is still not the same species, not exactly the particular against the particular species, so the efficacy of the antivenom might be slightly lower. Yeah, okay, and hence the dosing part you may have to adjust it accordingly. But there are also cases, for example, like because sometimes in the particular area, like for example in Indonesia, where they have this antivenom that is developed against 3 species in Indonesia, but when you have other species, okay, that causing the snakebite envenomation, this antivenom may not be useful or it may not be effective. Yeah? But without neutralization study, the clinician may tend to actually just use one antivenom for all the snakes in the area. So that could be something dangerous. Yeah. So the same goes for other species, for example, like king cobra, Prof. Neil just now mentioned. Yeah, okay. In fact, it is a complex, a species complex that's the Ophiophagus hana, king cobra, so widely distributed in Asia. So maybe there are differences, yeah, in each of the population, and the venom composition and antigenicity different as well. But the whole world has got only one king cobra antivenom, yeah, produced in Thailand. Yeah? So you can imagine that the effectiveness of this antivenom could be different, yeah, across different areas in the world. Okay, so sea snake antivenom as well. So this is some evidence to show cross neutralization. And for pit vipers is even more complex, yeah, because there's so many types of them, so many species of them, vipers and pit vipers. Yeah? Okay, so I think that the more of this, we can discuss more about this, but maybe in the panel discussion later. Yeah? So with this, I thank you. I hope this short introduction will give you some benefits. Okay, thank you very much.
And now we're coming up to our third speaker, finally a vet, Dr. Melissa Au, who works in Gasing Veterinary Hospital since she's graduated from University Putra Malaysia in 2010. And then 10 years later, she passed her European Master in Small Animal Veterinary Medicine in Neurology She also likes veterinary emergency and critical care medicine and had encountered and treated dogs with cobra envenomation, which need critical care. Without much further ado, I would like to invite Dr. Melissa to give her talk on snake bite management and challenges in veterinary practice in Malaysia. Dr. Melissa, please. Okay, Prof. Neil, Prof. Das, are you with us?
Yeah.
Okay, there are some questions that I think you already answered it to us. Would you mind from Nawa Nita Rupan?
Yeah.
Yeah, would you would you answer that about the wild Burmese python and all that?
Yeah. Okay. So the old records in the literature could be based on introductions, maybe unsuccessful introductions, because it's more of a seasonal forest species. So there were old records from up north and no recent records. So it's not in the fauna of Malaysia anymore. But there could be some escapees setting up populations because it's a very popular snake.
Maybe I would like to also ask Prof. Neil about that Burmese python and also python in Malaysia. Are they considered to be totally wild now, or some of them are also being captured for food items? I know in Sarawak, you could find sometimes people selling pythons as a food source.
So there are wild populations in the forest and also in the cities. They stay in the sewers. And I think they're more common. Reticulated python is more common in the cities than in the forest. In fact, all these years, I've found only one inside a forest, and mostly crossing the road in Bandar in Brunei or in Sarawak, and many captured from people's houses, also in Singapore. But the other species is Breitensteini or Brongus mai. They are forest species. They would not find them outside. And you mentioned trade. Yes, big problems here. A python that meets a human being is food. So although they're protected, they have in this state there are two levels of protection: protected and totally protected. So although they're totally protected, they are still hunted, and there's people who are caught. They are taken to task. They are sent to jail, fined, all that. But culturally, it's part of the diet of many tribes. So a lot of things need to be done before any conservation can happen, education, etc. Yeah, these are issues with wildlife in general, and when people hunt them for food.
There was a recent post about finding a very big reticulated python during surveys, and these snakes really protected, or should not be killed, or how should they be handled?
You're talking about snakes in general or reticulated?
The recent find of a very big reticulated python in Sarawak during one of the surveys done, and apparently it was killed. How should one do when
Well,
encountering such a
I don't know
big
about
snake? Yeah.
I don't know about this specific case, but when there is a snake which is found, encountered, most of the time it ends up dead, which is a very sad thing for most conservationists or zoologists, even citizens. But what is the option? The best option is to find out where it came from and release it there, which is difficult because big pythons are actually found in the gutters. Secondly, according to the legislation, it's illegal to kill snakes. So people do it, but don't face the consequence if they don't eat them, because they're kind of a bit lax on the enforcement. And the third issue is where do you put it back when we don't know where it came from? Sometimes we do a Google map search and look for the nearest forest. But there's been some studies putting transponders or transmitters on snakes, which are being reintroduced. And apparently when you put them in a random forest, the snakes are confused and eventually they get run over, they get displaced, they get totally confused. Eventually they die. So unless we have a lot of knowledge, we cannot handle conservation action properly.
And what about the interaction with pets? As you mentioned, these snakes are quite cosmopolitan now and can be found in our our house, around our house, and our pets. Sometimes, for example, dogs or even cats, has been reported to be so-called attacked by the big snakes or snakes, and some of them were actually injured as well. So how should we sort of like deal with preventing or protecting our pets against these snakes?
Okay, I would say most of the time the culprit is the Reticulate Python. And if it's a population in the city sewers, their diet for sure for mid-sized animals would be big rats, but those gigantic ones for sure their diets dogs and cats. So in a way they probably keep a control on dogs, stray dogs and stray cats. But when they start consuming our dear pets, it becomes an issue. So keeping our dogs and cats within a compound, I mentioned naive prey. Huh? They are not adapted either morphologically, behaviorally to predation. Huh? They're used to pampering. They're not used to an animal going after them to eat them. So, so keeping them secure where snakes have been observed, keeping dogs, especially at night, confined. Even in urban Kuching, there are farms which are raided by pythons, which eat up chicken, cats, and dogs that are out at night. Huh? So snakes are active at night. This is the time when we need to ensure the pets are indoors where large snakes are known.
Some of our pets are very inquisitive as well, and they tend to try to attack or play with these snakes, and some of the snakes are venomous as well, and they get bitten. So that was the issue that Dr. Melissa is trying to bring up during her presentation, I think.
Especially cobras would bite or even spray venom on the eyes of cats. That
so this animal can also go blind with the spray of for example, the cobra?
Yes, there's some studies on that. If you Google, you'll find ophthalmia, snake venom on cats and dogs, and of course the treatment's similar to that in humans.
Just irrigation and
irrigation
clean
of the eyes as soon as possible, otherwise loss of sight.
Okay, is Dr. Melissa ready?
Yes, I'm so sorry.
Yeah, can you share your screen?
Yeah. Can you guys see my screen and me and hear me clearly?
Yes.
Thank you. I'm so sorry. My Mac suddenly decided not to work when it's my turn. Okay, thank you for the introduction and thank you for handling the hiccups just now. Okay, so I am a veterinarian, but I'm not an expert in treating just snake bites. Whatever I'm presenting now is just from my experience, and it actually works to save pets that bites by cobra. Okay, so if you have any comments, just feel free to ask in the question and answer box. So I'm going to present about snake bite management and challenges in veterinary practices in Malaysia. Let's let me play this. All right. As you can see, most of the snake bite cases presented to our hospital, I'm not sure about other veterinary clinics and hospital, most of the time we get snakes like bite the dogs more than the cats. So most likely the dogs will be very more curious than the cats, and maybe I think the cats is a little bit smarter not to go towards the snakes, you know. And also the most common snake bite I encounter is cobra, and the less common snake I've ever seen is python. But maybe the rest of the snake, where the owner gave us a photo and stuff like that, we might not be able to identify because we are not an expert in this field. So in this slide shows that in the past 10 years, these are the amount of snake bite cases presented in our hospital. And all of the cobra and venomation or dogs that have been spat by cobra is all in dogs. However, in 2 of the python's cases that we saw is both in cats. So there is an increment in cases throughout the years, and there are some reduction in cases in 2017 because we are unable to get cobra anti-venom. So most likely, most pets owner actually move on to other clinics to treat their pets. So what are the clinical signs when you have a dog with snake bite, especially cobra? I'm explaining more on cobra because this is what most of the cases being presented to our hospital. So in this dog, it's been bitten by a cobra. It has severe angioedema with swollen face, lips, the tongue, and the eyelid is not really swollen in this photo itself. And this is actually a Labrador retriever, but it doesn't look like a Labrador retriever now in this photo, okay? So the next most common clinical signs we can see is Blepharospasm, which the eyelid of the dogs will close tightly because of pain due to corneal ulcers. And these are mostly caused by the spitters, like what Prof. Neil has mentioned, by those cobra that spit the venom into the dog's eye. And we most of the time, we will get very diffuse kind of corneal ulcer rather than those pinpoint corneal ulcers that we saw in cases of injury due to like grooming and stuff like that. So another most common clinical signs I've seen is dogs presented with difficulty in breathing, and eventually some will turn stuporous or comatose. And of course, most of them will get snake bites, wounds, as shown in this case. This is from a dog. His name is Cluso. He has been bitten by snakes several times that I lost count. And this is one of his worst encounter with a cobra, which his skin turns dark and it's turned necrotized, and eventually become a very, very deep wound. But he survived this. He survived all the cobra bite wounds, and he's one lucky boy. And most of the snake bites we will see is mostly in the muzzle, the legs, especially the front legs, the neck, and the shoulder, and also the tongue. So how do we treat snake bite? So first thing, we have to identify the snake for sure. But of course, for me, I'm not an expert. Sometimes they can give us photo like shown in this slide. I may not be able to know whether is this a cobra or not. So most of the time, it will depend on the clinical signs. And I would actually monitor if some dogs doesn't show any severe clinical signs with only wounds. I would try to refrain myself from giving any anti-venom until I'm quite confident that this is due to a cobra envenomation. All right. So we also need to understand that not all snakes are venomous. So it's not necessary that when the owner comes to you and say, my dog has been bitten by a snake, we have to be very calm, don't panic, and ask the owner accordingly what kind of snake it is. Do they have a picture? It is even better if they can bring the snake over to us to have a look at it. So at least we are pretty sure what type of snake and how are we going to deal with it. So we only give appropriate anti-venom after the snake is identified. So of course we have to put intravenous drip no matter what, because especially if we cannot identify the snake, we're going to monitor the dog. It's best to have an intravenous drip and an IV access. If anything happens, like the dog can't breathe, we actually can do some emergency procedures for that. So if there's a wound, we have to clean the wound. And if there's any corneal ulcer, we have to flush the eye vigorously with normal saline to ensure we actually flush out most of the venoms out. All right, if a dog is apneic or they can't breathe because of neurotoxin, intubation and manual ventilation are needed most of the time. And I have ventilated a dog which has bitten by a cobra. He's a Weimaraner. His name is Paco, I still remember that. I ventilated him for almost 16 hours, but he survived. So if we try hard enough, I feel we can help the pets. We just don't give up, okay? And the biggest challenges in Malaysia is we don't have a good guidelines for snakebite management in veterinary practice. And as Prof. Neil has mentioned, it's very, very little like journals that we can actually refer to, especially in Southeast Asia kind of venomous snake. So, and for us, most veterinarians, I'm not sure about the rest, but for me, I am really not familiar with snakes at all for those species. So I would depend on people like herpetologists, people like Norsham sometimes will help me to identify those snakes through pictures and stuff like that. So, and for any snake bite kind of like treatment, time is of essence. I need all the vets to know. So we actually need to get appropriate anti-venom to treat the pets. So, but most of the clinics do not have anti-venom or are not aware that we actually can get anti-venom in Malaysia. So this is something, is a challenge to treat snake bites. So the next slide, I would like to explain about the treatment for Cobra bites. I would actually divide it into 3 parts, which is one is like a Cobra envenomation, where most of the time they really get in the venom, they have clinical signs. Some are being spiked by Cobra and some are actually presented comatose. So those with Cobra bite envenomations, you have, if it's a true envenomation, we have a swollen face, you have a wound over there that is obvious, you know this is a Cobra when the owner bring in the snake or they took a photo of the snake. So I think that we should not delay treatment. We should actually start the patient on anti-venom, all right? But in our practice, because we do not have anyone to guide us, I mean, the only one guiding me is actually my boss, which has a lot of experience. And he actually has this little treatment protocol that we always give dexamethasone before we give anti-venom, because we are very, very afraid of any anaphylactic shock. So we give dexamethasone and we actually take 1 ml of anti-venom and we give it really, really slow IV within 10 minutes. And the remaining 9 mils of the anti-venom, most of the time we actually reconstitute into 500 mils of normal saline or even 100 mils of normal saline, and we let it infuse at maintenance rate for about maybe 4 to 5 hours. So most Cobra bite wound in cases that, I only have 2 cases, we actually put intra-lesion where we put the anti-venom around the lesion. I'm not sure how much that can help, but maybe Dr. Khaldun can share a bit in humans, do they use that? Because I tried that, but I don't see any difference putting intra-lesion and not putting intra-lesion that really help in the wound. But most of the time, I will really lavage the wound very, very vigorously. And we actually put a lot of antibiotic cream and give oral antibiotic or intravenous antibiotic to treat for secondary infection. And this is, of course, Clouseau, the famous dog. You can see the wound progression that they can be very simple, like very dark color. Maybe you don't even know it could be a Cobra envenomation. And slowly it will progress into a very severe, deep wound with necrotized skin and subcutaneous tissue. So, and this wound will take about a month to heal. So we just have to be very patient with these kind of cases. And the owner has to be quite committed and understand what is the outcome of all these Cobra bite wounds. And some cases of the dog actually presented stuporous, where they are not conscious. And most of the time they can't breathe. And we have to intubate when it actually comes, when they walk into our clinic. And ventilation is extremely important, because if you do not ventilate the dog can die of hypoxia. And I think that's what leads to them become comatose. So in some cases, I do actually administer mannitol within 20 minutes within this dose to actually helps to, reduce any possible cerebral edema due to hypoxia. So a close monitoring is needed. Like I told you, I have ventilated the dog for 16 hours. Of course, I take turns with my colleague. So you might need a hospital that actually can, monitor the dog regularly to do that. So another thing is we can use modified Glasgow Coma scale, which is very useful in this kind of cases, as we can actually give a prognosis to the owner depending how the dog responds to the treatment. But most of the time, of course, before giving mannitol, we have to give anti-venom and all sorts of those kind of things. And the mannitol, most of the time, I will give it in a different IV catheter. So the anti-venom will be in a different IV catheter and mannitol will be another IV access. So we have 2 IV access, so I do not disturb the infusion of the anti-venom. But most important, you have to administer anti-venom. But if you realize that the dog is completely stuporous, the modified Glasgow Coma scale is not good, I think you should, and no harm that we actually start on mannitol to try to reduce any possible cerebral edema in the brain. So for dogs that's fed by cobra, so they most of the time they have blepharospasm. They will come in very, very squinty eyes, swollen eyes, and owner will always panic because they say the dog has been fed by a cobra. But as a vet, we don't panic, of course. We need to stay calm and just assess the dog first for any bite wounds, any difficulty in breathing before we actually, start to administer anti-venom. Because for this kind of case, most of the time I do not administer anti-venom. If you give a very good ocular lavage to actually flush out all the venom, and you do a fluorescent stain, of course, to assess how bad is the corneal ulcer. But most of the time in the first day, you will not be able to do a fluorescent test because the dog will be shutting his eyes so tightly that you can't even access the cornea properly. Probably you can do it the next day. But you have to really lavage the eye, put a lot of antibiotic eye drop like chloramphenicol, gentamicin, or ofloxacin. And besides that, we also use we actually harvest the plasma from the dog's blood. We actually drop into the dog's eye every hour. And that works very well in our practice. And most dogs within 24 hours, you can actually see the eye open. And that's the time I actually assess, how deep is the corneal ulcer. And I would say out of 9 cases, I will not give cobra anti-venom for this kind of case. But there's only one particular case that we did give anti-venom because there is only a corneal ulcer, but the dog's face actually get very swollen and the swelling didn't subside. So we actually gave the anti-venom and everything goes down. So you have to still monitor the dog. I would say probably, even though the dog presented with just a speck by cobra, but I usually let the owner stay in the hospital for at least half a day to ensure there's no angioedema signs, difficulty in breathing before I actually send them back home. And as I said, not all cases actually needs anti-venom. Only for cases where it's fed by cobra, okay? So the prognosis is generally good if we give appropriate management and treatment as quick as possible, okay? Especially for cases with neurological and respiratory symptoms. And most of the dogs will survive. There's only very minority of the case that from our place didn't survive because the owner came quite late and the dog was really bad and comatose and cyanotic that we have to resuscitate. But do not give up. Even they came in with very blue tongue. If the owner told you it just happened within in 15 minutes, maybe from their home, just try your best to intubate the dog. Make sure the airway is patent, try to resuscitate at least 20 minutes. We just have to try as best as we can and put in the anti-venom because the cases are quite rewarding that we managed to revive the patient, like the case of Paco, and the dog actually survived. It's very rewarding, tiring, but very rewarding. So what can we look in the future for veterinarians for snake bite management? Hopefully we can form an interest group and slowly, we can get a guidelines for snake bite management in pets. Okay, like in Australia, they already have a guidelines for any venomous snakes for them, but not in Southeast Asia. But this is my little interest. If we can form a group for that, we have Dr. Taxa in the first webinar. I mean, we can get people around us as a group, as a team, as veterinarians. I think we should form our interest group in this field. And also, it would be very good. We have herpetologists, medical doctors with very good, knowledge on snakes ID, and medical doctors with very good guidelines on snake bites in humans that we can use as an example, or at least as a, as like a guidelines for us to form our treatment protocol in the future. So I understand that most vets practices do not have anti-venom. So it's difficult when an owner goes to one vet clinic and they end up being sent away to another clinic just because that veterinary clinic doesn't have anti-venom. But I personally think that it's a little bit a waste of time. So if in the future we can get like a veterinary hub that we can call this specific hospital to let them know they have this patient over there that been bitten by a snake, we probably, you can send it over to us, give us a call. We can actually loan the anti-venom to you. just save the pet first, and you can return back the anti-venom to us when you're ready. So my presentation is really short. So now I'll pass the ball back to the moderators. Thank you.
Thank you, Dr. Melissa, for your sharing. The hiccup that you experienced actually gave us a chance to answer some questions from our participants. And moving on to our next speaker, Associate Professor Dr. Scott Weinstein. He is currently a clinical toxicologist at the Women's and Children's Hospital, Adelaide, and Associate Professor with the University of Adelaide School of Medicine and a Fellow of the American Academy of Family Physicians, besides being a venom researcher and a family physician. His academic trainings include general and field herpetology, medical microbiology or immunology, biomedical sciences, and comparative religion. His experiences include treating snake bites and marine envenoming, as well as poisonous ingestions, characterization of aberrant toxins present in snake venoms, isolation of antimicrobial components of venoms, pharmacological studies of venom toxins in the red blood-brain barrier model, and iontophoretic investigations, and antigenic relationships among venoms and secretions of blood of non-front fang snakes. Dr. Scott, he is also a faculty member of the recurring ANSEM Symposia. He has also contributed to more than 100 peer-reviewed journal papers and 4 books in toxinology, herpetology, and clinical medicine. Today, Dr. Weinstein will offer some casual observations about envenomed veterinary patients from a human clinical toxinologist's perspective. Let us hear from Dr. Weinstein.
I'm looking to come up here.
Yeah, you're on.
Yeah, I know, but I can't seem to bring myself up to it. But anyway, well, thanks a lot for the introduction. That was quite a bit. I'm really here for just a couple of comments. That might be quite a bit less than was described, but I appreciate that very much, and it's a pleasure to be here. This is really a perspective just that I'm offering on somebody who treats bipedal primates, i.e. us, and I've been asked to consult on quite a few cases of companion animal envenoming. At last count, I believe it was somewhere between 60 and 70. So my experiences are largely between the United States and here in Australia. As you've heard from a couple of the very nice talks that were given, there are far less data from Southeast Asia on companion animal envenomings. And that is really unfortunate because I suspect that they're a lot more common than we realize. But I just want to emphasize a couple of, as was stated, a couple of casual points. That is, just to emphasize, I'd like everyone to be aware that sometimes what we see in humans is sometimes expected in a companion animal or an agricultural animal that gets bitten. And because the literature that's consulted, there'll often be an expectation that we're going to see the same type of effects. And there's some examples, particularly in Australia, of how incorrect that can be, because the most common species of snake or group of snakes, there are 9 species here in the genus Pseudonaha, which are the brown snakes. And of those 9 species, there are really 3 or 4, particularly Pseudonaha textilis, the eastern brown snake, Pseudonaha mengdeni, which is the western brown snake, Pseudonaha nucleus, which is the northern brown snake, and Pseudonaha aspidoranca, which is the saddle-nosed brown snake. These are involved in the very disproportionate number of envenomings, including in companion animals, and particularly fox terriers, dachshunds, border collies, and Jack Russells. And in other words, this the dogs that most commonly will go and put their nose pretty much anywhere out of curiosity. They very often get bitten on the snout. And the interesting thing is in brown snakes, the most predominant toxin that we see are really what are called C and D class procoagulants, which very much are have an amino acid sequence that is highly homologous with human factor 5 and 10. So we see in humans a notable, severe, total defibrinating coagulopathy with, of course, also thrombocytopenia and a great bleeding risk. But yet in companion animals, we do see a coagulopathy, but we also see a profound, rapidly developing, presynaptically stimulated neurotoxicity from the most toxic snake venom toxin known so far, which is textilotoxin, which is a pentavalent presynaptic, again, heteropentameric neurotoxin that in humans is clinically insignificant. And this is at least rare, I should say. And we don't know the exact reasons for this, except there are several beliefs. One is that, of course, canines and felines, the most common animals that we see bitten, are highly susceptible to this neurotoxin. They are knocked down as if they're shot by it very often, particularly cats. And then the other idea is because humans now get antivenom more rapidly, we may be interfering with that sooner, but there's a problem with that because, as I'm sure many people here might be aware, presynaptic neurotoxins are very hard to neutralize with antivenom, especially if they've started their action, because as you heard, from Dr. Tan, they will very rapidly block neurotransmission. They will degrade the motor end plate to an extent. And then, although there is some conflicting data about that now, going by what we do know, it will usually require a period of intubation for the motor end plate to recover and to wind up getting appropriate transmission or transmission again. But yet in humans, we very rarely see this with brown snakes, and canines and felines are affected severely, profoundly, very rapidly by it. So what Dr. Melissa just pointed out about intubation is life-saving in some elapid bites by cobras, and in other parts of the world, mambas and coral snakes and so forth, and sea snakes. Very unlikely, obviously, with pets. But the most important thing is the most common elapid bites, if there's the presynaptic neurotoxicity, as we see in Australia and sometimes in other parts of the world, that is a critical life-saving phenomena. I can tell you also the other thing that brings us to is that in Southeast Asia, most of the time you're going to be talking with postsynaptic neurotoxicity. And so that may not be as critical of an issue because you can give, as you heard, things like neostigmine, acetylcholinesterase inhibitors that are going to actually help even in the absence of antivenom. So I think one of the strong things I want to emphasize is, although you may need to consult the human literature on clinical toxinology in order to facilitate management of envenoming of a lesser documented particular envenoming in an animal, you have to be very cautious. Do not expect something to evolve that may not, because of these differences in the pathophysiological manifestations between the species. The other part of it is that antivenom availability is a critical issue because, obviously companion animals deserve to receive it when they need it, just as we do. Of course, the expense is something to be considered. Just a couple of weeks ago, I was asked to consult on a bite in a fox terrier, and it was a very profound bite by a brown snake. The animal was completely paralyzed. There's still, of course, the coagulopathy. So we wanted to treat with a vial of antivenom, which would have, which costs the owner roughly about $1,200. And then the vet and his assistant were extremely dedicated. They were intubating and bagging this dog for some 12 hours. And I could hear the owner on the phone, because this was in another state, who said, well, mate, talking to the dog, well, mate, you better have 9 lives like a cat, because the next time there's no antivenom, because it was obviously going to, the vet bill was going to be several $1,000s. So this is something to consider, but I do think there needs to be availability of a central distribution point for antivenom. I know that in some places that already exists. it's important because the effect on animals can be so profound, so rapid that the need is somewhat immediate. The other issue is with venom ophthalmia that was mentioned briefly as well. That is not uncommon in other parts of the world and in Southeast Asia included. And the issue there is, of course, getting adequate irrigation with a companion animal could be quite difficult because of the resistance to the treatment. So sedation, anything that can help the irrigation and as quickly as possible should be instituted. Putting a Morgan lens in a dog would be extremely difficult if it's possible at all. So you'd have to really rely on almost a continual drip or a thorough continuous irrigation. I might add one more comment on this, and that is, I know it's a common practice in Latin America and Southeast Asia to give dexamethasone and even prednisone, corticosteroids before giving antivenom, but we have very little evidence to support that, largely because, I'm sure many of you know, the intravenous peak of activity of giving that IV is about an hour, minimum 45 minutes. So what is it really doing when you're trying to prevent an acute response, which is normally you would give adrenaline? Now, there's very little evidence to support any of that in veterinary medicine. So as was stated, there needs to be more studies done on that. In humans, I do not give steroids of any kind. I use adrenaline, and we only pre-treat when we know that there's a greater likelihood of hypersensitivity of previous administration of antivenom or of a particularly susceptible patient, or giving an antivenom that is quite dirty. Some of the antivenoms in parts of India, parts of Africa, have a degree of contaminants, usually serum proteins, and you wind up seeing a febrile pyrexia, like a white ghost response from transfusions, and adrenaline often gives that. And that is the other point. Most of these hypersensitivity reactions are not IgE mediated, which is why skin testing is useless. These are adverse reactions due to complement activation or due to the contaminants in a commercial antivenom. I doubt that it's very different in companion animals, depending on the nature. I've been involved in 2 pet primate envenomings that were not snakes. They were scorpions. And in those particular situations, one was fatal, the other one wasn't. And in one particular case, the monkey did have a notable, it was a capuchin monkey, Cebus, genus Cebus, that had a notable hypersensitivity reaction, required adrenaline. The other thing is quickly on to non-front fang colubroids, which was mentioned as a particular interest of mine. We have a very unusual example of a magnified example of what could happen with Professor Das was mentioning, with an invasive species on Guam, where we have Boiga irregularis related to the Boiga in Southeast Asia, not the same species, but related to it, the brown tree snake, which was introduced from the Admiralty Islands off of PNG during the very large transports that were occurring during the Pacific theater of World War II. And they got sneaked into Guam. They were introduced. They essentially destroyed the bird populations. And in recent years, what they've been doing is preying on pets, attacking human infants occasionally, definitely with predation attempts, and also feeding on pet food, which is a very unsnake-like behavior. And the treatment of pets by them also involves physical constriction, as it does for the human infants. I would pose to you that the effects we see in human infants is not really clearly assignable to venom. It's more assignable to physical trauma, thoracic trauma from very, very powerful constriction. And we see that in small dogs and cats there as well. So as Professor Das also pointed out, the physical effects of python attacks and so forth can't be ignored. They are equally important. I think those are my main comments, is to be aware of these divergence between the effects in humans and the effects in companion animals and agricultural animals. And I think the most important thing to emphasize, really, as just not to bore you, just as a couple of final comments from myself, is that there's a real need for formal study of this and for some harnessed type of data that really can be analyzed and then used to develop solid evidence-based guidelines. Because I would say that we do have guidelines here in Australia, and in the United States, we have some for rattlesnakes. But for example, there's a few cases in the US by the harlequin snake or the eastern coral snake, Micrurus fulvius, where we have megaesophagus that's developed very unusually, you know, from a couple of coral snake bites. There's also efforts in the US, excuse me, to use a rattlesnake vaccine, which was developed by one particular company. And there's very mixed data on that. Those data indicate most of the time that it's quite ineffective. There are some other data that suggest that it might have an efficacy in some animals. And it's been attempted to be used. It was made from Crotalus atrox, from Western diamondback rattlesnake venom. To my knowledge, I believe that's the only venom that's used. So it'd be technically a monovalent vaccine, if you will. But it's been attempted to be used in equine patients as well. And my understanding of that is it's totally ineffective. But whether or not it works in dogs is up for grabs. And my own opinion is it probably doesn't. Unfortunately, there's a history of developing vaccines in humans, primarily against Protobothrops flaviviridis on Okinawa. That was a habu venom. That was a terrible failure because the people receiving the vaccine developed terrible local severe ulcers and severe local effects, and it was discontinued. So I think vaccines against venoms are unlikely for the reasons that they're unpredictable in their response in the host, and they are not really, shall I say, reproducible. And so I think that vaccines in pets is another thing that is worth investigating, but we don't have good data that any that are available now are really of any use. And so I would say, by all means, study this and record your data, because as was stated, in Southeast Asia, we have very, very little data on the effects in companion animals and agricultural animals from the snakes that we know are medically significant to humans. And publish what you've got. Publish it. Don't just keep it to yourself. The rest of the world wants this information too. So I strongly encourage everybody listening, please keep track of these type of studies, even if they're individual observational studies. Do some retrospective studies in the veterinary clinics you're in. Do some of that work and harness this data together, so it will also give some idea how to develop maybe the initial studies to develop guidelines. So that's pretty much what I had to say, and I'll be happy to answer some questions if they come up directed to me. Thanks a lot.
Scott, I would like to add a bit on that notion of you saying that, you know, because of the issue of price, I mean, they have to pay for this can be a very big stumbling block for some people to take life of animals as secondary rather than primary family member, as you said. So perhaps that is another area that maybe the veterinary service can maybe put forward to increase the awareness and maybe allow more people access to the services to look into these so-called pets or companions, the four-legged companions. The other part about anti-venom is, or venom is the specificity. As we know, the Tropidolemus as well may not affect human, but as much as it will affect the avian or the murine group. I mean, mice and birds. So people keep birds, people keep big mouse or guinea pigs as pets, and they might be affected more than what we see in humans. So that could be another, as you said, some things that we see in humans may not be translated to what we see in our animal companions. The other part is Dr. Melissa mentioned about the practice of giving antivenom intramuscularly. Maybe you want to say something about that. Obviously, we don't practice anymore in humans.
No, and
yeah.
That's actually in fact, I meant to say something about that. You just reminded me. That's a very important point that that Associate Professor Khaldun just mentioned there is that the fact is that we have very strong data that if you want to get rapid interstitial absorption, in other words, a broad-based distribution of the antivenom, then we really should be giving it intravenously. It doesn't matter who or what. As a matter of fact, I just dealt with a redback spider venom, that's Latrodactys hasselti. We see many of them this time of year here in Australia and black widows in the US, where there's still an emphasis to occasionally give the antivenom IM. And I can't tell you, from specifically objective data, there's a difference, but believe me, clinically there is. It needs to be given and get into the intravascular compartment as quickly and in a large dose as possible. Because remember, I saw a question come up before that I'm going to address briefly. It said, has the half-life of the venom been studied? But venom is not one component. Venom is hundreds of components. We don't really know the half-life of almost any of them. So we don't know if they're processed through cytochrome P450 complex. We don't know if they're just renally excreted. We don't know if they're activated after a first pass metabolism. These are all different components. Some act synergistically. So the antivenom better be in the blood, because otherwise you can have these components, different molecular masses being absorbed at different rates. We don't know how they're being exposed to the antibody. The best bet is to hit them all at once, as much as you can, as early as you can, which as much as you can, flood it. There seems to be a terrible, I've been having a war of words, as some people know, in Australia with a group that believes that one vial of antivenom treats everything in Australia the same way. We know this is nonsense, because the severity of envenoming is different from person to person. And I don't have to tell anybody here, if you have a dog or a cat, just like a human, with a pre-morbid condition, a cardiovascular condition, hip dysplasia, it can't walk around as much in a shepherd, or a Great Dane, and they're 9, 10 years old and they're starting to limp. You think that's going to have the same response to an envenoming as a perfectly healthy 3-year-old dog? I don't believe it. So you need to give more antivenom earlier. And that's the way we treat this. We get in quickly and treat it early. That's the way you get on top of this. Once the envenoming takes hold and you get multi-organ pathophysiology in process, your efficacy of treatment is going to drop.
Yeah, I think that's where the last bit where Dr. Melissa was suggesting a closer look into establishing the antivenom storage stocks, and also, of course, the guidelines for veterinary medicine here in Malaysia or the region. I think Taxa in QSMI under Prof. Prof. Visit leadership had actually already established quite a good service for veterinary medicine regarding envenoming. I think we can use their help as well to build up the guideline for the region. And I totally agree. Some of the points that you put up about species will require a study for that particular country itself. You cannot apply one guideline from another country to the same as your own country, because the species is different. The venom composition is different. Obviously, the efficacy of antivenom use also will be different. So this will bring us to the next session, which is the forum. I think I will pass back to our moderators to conduct the forum. Thank you, Scott.
Thanks a lot.
Thank you, Prof. Weinstein. That was absolutely amazing. I mean, I just fixed on the screen and just listening to you, and I know other people also will rewatch it, because that was like an absolute good insight. And we know that we lack of so many things, and I felt that I'm not a vet, but I understand the vet are so busy. That's why I'm trying to organize this, because I feel that we need to come together, the medicine, the human medicine and the animal medicine. So I'm glad that all of us, this is like a second networking, and I do hope some feedback from the participant later on what else can we do to go forward. So back to the Q&A. I think this one, there are some of them already been answered. So I think there's one for Dr. Tan that I would like Dr. Tan to answer. Yeah, Dr. Tan, are you with us?
Yes.
Okay, just one question by Jia Wen Tan. Can we use the strongest anti-venom to treat envenomation from less venomous snakes of the same species? Will it cause any side effects or etc.?
Sorry, where is the question?
If you see in the Q&A box, there's one,
Yes,
yeah,
open.
Jia Wen Tan. Good day, Dr. Tan. Just curious, can we use the strongest anti-venom to treat?
I think can everybody turn on the video?
Yeah,
Okay. Jia Wen Tan.
yes.
Yes.
The strongest antivenom to treat from a less venomous snakes of the same species.
So what? I think if you are treating the if you are giving antivenom, the primary concern is whether, of course, if it's indicated, then the primary decision should be made based on the species specificity. And I am not sure what it means. What he means with strongest antivenom. I assume that there's only one antivenom, is it? Are we comparing different antivenom products? So
I think it's a misunderstanding. I think the
yeah,
question is blurred. I think the understanding about what is the strongest antivenom is not about the strongest antivenom. There's no such thing as strongest antivenom. It's about the appropriate antivenom for the appropriate species, as what Prof. Scott was mentioning just now. So it's not about the strongest antivenom. It's about the appropriate and
yeah,
adequate
yeah,
antivenom for that particular species or producing
yes,
the envenoming. Okay.
yes.
There's one question looking at, instead of using anti-venom, is it possible for us to use alternative drugs like Chinese traditional drugs or neuroprotective drugs to alleviate the severity of case? Who wants to take this?
I think at the moment there's no evidence to suggest other than antivenom works with envenoming syndrome. Prof. Scott, maybe you want to add?
I know that there's a lot of strong, compelling reasons regionally for people to reinforce their confidence in using local remedies or using tried and tested approaches to local healers. But look, you know, the basis here in clinical toxinology, the whole effort is to make this evidence-based. We have absolutely no evidence that they do anything to alleviate or to neutralize the effects of envenoming. And the most important thing to emphasize there is the last thing you want to do is waste time trying things that don't have evidence to support them in lieu of doing what we know actually works. So I strongly discourage that.
Okay, there's one question. Is there a fatal cases of colibrid bites in Malaysia? How do we go about it symptomatically? I'm not sure this is question for human or pets.
Yeah, I can't help but jump in on this.
Yeah.
Okay, I mean, I'm just finishing the second edition of our book on this, so I can't help it. Look, there's no fatal cases known in Malaysia in humans whatsoever, but that doesn't mean that there can't be, because there are a couple of species, not of colubrids, but of netricids, which means they used to be a subfamily of the colubridae. They're now the family Netricidae. Those are redneck keelbacks. And redneck keelbacks have caused fatal envenoming. So they can kill people, and they can kill dogs, and they can kill cats and other animals, but not yet. Unfortunately, the sad part of this, to make a long story short, is that Japan used to have an antivenom against the congener, which is Rhabdophis tigrinus, the tiger keelback. But I can tell you through checking this through great efforts, because it's not easy, I have really confirmed that Anti-Venom is no longer available. They're not making it. There's a small archive group of Anti-Venom ampules that were made that are still used, and they're experimental. So even if you're in Japan and you have a potentially fatal human bite, you have to go through a paper process to get access to it, which can take quite a bit of time. It is very unlikely Anti-Venom will be available. So it's symptomatic treatment. And if anyone or any pet is bitten by one of those and it's confirmed, they need to be put into an intensive treatment regimen, which is too extensive to get into now. So the answer to the question, though, is no, not yet, happily.
Thank you.
In humans, no, not in, well, in humans, not in Malaysia, but there are cases in Vietnam and Indonesia, which cause severe bleeding. And some of it are recorded as death as well from the bleeding.
Yes.
Animals, I think we have no documentation about from the Rhabdophis envenomation.
Dr. Melissa, there's one question. I think you may want to take it. Will skin sensitization test with perhaps 0.1 ml of antivenom help to find out whether the dog can take the antivenom?
Okay, now I again I have to jump in because I specifically said a few minutes ago that you don't do skin testing. Okay, that's not going to be useful. The main reason, again, is because we think we have good evidence that the hypersensitivity to antivenom when it's present has nothing to do with IgE. So there's no point in doing a skin test. It's an adverse side effect, very likely of complement activation. Again, like I compared it, it's a similar disease to what we see from white ghost febrile pyrexia. So what you would do is you would give adrenaline if it occurs, but you don't do skin testing. It's not predictive of sensitivity at all. It could come up positive, the person's actually not sensitive. It could come up negative and there is sensitivity. So it's useless. So don't do it.
Thank you. We are running a bit of time. Are we going to pass it to the chair or?
No. Continue. We have a few more interesting questions
Okay.
to answer. Yeah. Go ahead.
Dr. Wyman, do you want to pick up from me? I mean, to ask about the question and all that. Do you want to?
Yeah, it's just a couple of questions on the dosing references by Gideon. He's asking on regarding the dosing reference for the anti-venom on animals and any reference values regarding the dosages to be given depending on the animal. Anyone would like to take that up?
Well, I'm talking a bit too much here, but I would just say that those are very good questions. I mean, you know, I think those are very valid questions. Unfortunately, what we usually recommend, I could tell you here, for a brown snake envenoming, I recommend giving pretty much a human dose. We start with, you know, now here's where I get myself into trouble because we start with 1 vial. Now in humans, I start with 2 vials, but with a dog, because of the expense and the size, I'll usually recommend starting with 1 vial, but the question isn't really answerable because we don't have data supporting that. See? So I think those are very good questions that need to be assessed by evidence-based trials. Because right now we don't know. And of course that won't apply to the, let's say, Thai Red Cross antivenom. You know, we don't know, I don't know if Dr. Toxa has, you know, some data on what might be a good dose for a given breed of dog. You know, from one of the very good antivenoms they make there. But to my knowledge, that hasn't been ascertained.
Yeah, to pick it up on that is that the way that we give antivenom for even humans or human is purely what do you call looking at the signs and symptoms of response as well. So we just don't know different animals. Human is just one group of an animal, but in other veterinary services, that dogs, cats, they may have a different volume as well. body mass index, and so on. So it's a bit complex, and so that's why when we just pick one way of giving antivenom is going to be, I think, can be quite dangerous or inaccurate itself. So that's where the data from maybe Thai Red Cross, where they do provide antivenom for their veterinary service, can be a starting point, because we are quite close to Thailand, and we have quite similar species as well of snakes, and we can actually look at their guideline to have a look. But I think they also start with one vial, and they proceed as the symptoms progress, response to the antivenom. And I think that's what we did when Melissa contacted me for the treatment of the dogs, where we gave our antivenom to the dog. So we start with a lower number of vials as compared to humans. So we start with, I think, 1 or 2 vials, and we increase the dose as the response. So yeah, it opens a lot of research opportunity, I would say.
Yeah, definitely. And I think the vet have to do something about this. Wherever I can help, I will try to help. But yeah, Prof. Weinstein, another question for you from Atif Shah. I have a small animal clinic. Unfortunately, we don't have any antivenom available in my place. If I have a patient with snake bite, is there anything I can do in order to stabilize the animal before I refer to a hospital for further treatment?
Again, I think that's quite a good question. And the only answer I would say is it depends on what's bit them. So, so in other words, like I gave an example before of like, pseudonav brown snakes causing profound, rapidly evolving paralysis, and without intubation and vent, ambu bagging really, that dog or cat would be dead. It just would die from asphyxia. So I think the key factor there, my answer would be, it really depends on the nature of the syndrome that you're seeing, the envenoming. If you're seeing something from a crotaline viperid and you're seeing, bleeding, a hypovolemic, obviously you can do fluid resuscitation. You could try to even provide some albumin products if it's available with care, because the reason why we have to be very cautious about that is because without antivenom on board, you're going to put more substrate. If you give serum products, you could wind up putting, well, plasma, I should say, you're going to be giving substrate for the venom toxins to act and produce more disseminated intravascular coagulation in some cases. So the answer to that is conditional. What has bitten the dog? What's the predominant symptomology? And so if you're seeing an elapid bite and you're seeing the beginning of paralysis, I would not wait. The supportive treatment would be intubate and vent. And that could be life-saving for the animal.
I think we answered most of the question except one. Any studies done on the efficacy of each antivenom? That is kind of broad, but then
Maybe Dr. Tan can answer that.
Dr. Tan. Dr. Tan, are you with us?
Yes.
Thanks.
Hi. For all antivenoms and all snakes, no, much research is needed. It's just too complicated. You got to assess the efficacy, per antivenom products, and then per species. And you got to also consider about the potential variations in each venoms, and also the variation that probably comes with different products, or even batch to batch differences in the antivenom. So there's no such data, but research is ongoing. So I think more data is coming up.
If that's all the time that we have today, thank you, Professor Das, Dr. Tan, Dr. Melissa, and Dr. Weinstein for your sharing today with us. And before we end this webinar, we would like to call upon Dr. Khaldun once again for his closing remarks. Dr. Khaldun, please.
Thank you so much to our moderators for very good sessions. I think we have very interesting questions, and I think we have addressed some of them quite well, and it opens up a lot more questions actually, and this is very good for research, and I think we have quite a big responsibility now from the veterinary services to find the answers to all these questions. So I would like to thank to all our speakers. As I mentioned earlier, this is not the last program, and we are recording this. So if you missed the earlier part, you can always come back and review the recordings, okay? And do join us in our next session. So before we finish, I would like to also personally thank our technical support by Dr. Ruth Sabrina. She's our emergency physician, Rex consultant, and also the vice president of MST for a lovely session today. We had quite a very good turnout. We had several hundred responses, registrations, and participations. So all of you are actually able to obtain the so-called MMA CPD point by scanning this QR code. You can scan the QR code. And for everybody else who wants the e-certificate, please fill up this Google form. Maybe our moderators can share the link of the forms in our chat group, in our chat, so that everybody here can fill that response and obtain their auto-generated e-certificate. So I will leave this page for the MMA CPD point for several more minutes before we completely turn off the Zoom meeting. So before I say goodbye, I would like to ask anybody in our panel, our moderators, if there are any other last words before we say goodbye today. Norsham, anything?
I'm good. Any other panelists would like to say anything?
Waimun, anything to add?
Not from me, Todd. Thank you.
Okay. Anybody else? Our moderators, our panelists, Prof. Indranil Das, any last words?
Yeah, okay, all right. So I mentioned to someone in the Q&A about the resources. So I pointed out a book by Prof. Kal and some of us on identification. So you could download that huge document, or if you're lucky, you can, I think, bid in an auction or something. There's some some way to get a hard copy. And otherwise, a bunch of other
this
books.
book, right?
Yes.
Yeah, this book.
Yeah. And also this one.
This is available. This is available online. You can download it from the MST website. Okay.
And we look forward to the second edition of the Rear Fang Snakebite book by Professor Scott. I'm sure it will be much referred to classic. Professor Scott, do you want to say anything about your upcoming work?
Well, I'd love to, but quite frankly, I just got a plug, and I just, you see what's happening here is that some of the other panelists here are just trying to get greater amounts of required payback from me. That's what it's all about, see. So, but I'd like to ask a question. Why didn't Chu Hawk have a sketch of him on there? That's just a thought. That's all I have to say, essentially. That's all. Thanks for the plug. I guess I will add one thing, and that is that, yeah, the book is finished. It's just entered production, and it really does. It's a different book. It's just required an enormous amount of work. And I might add that a couple of the panelists here that I'm still seeing right here have been very generous contributors to it, not only with photos, but with information. So I have to say, I have to take a moment to thank everybody, particularly Prof. Das and Associate Professor Khaldun for for being so generous with very, very interesting and important information and images on that. And I can tell you that a certain professor from Oxford also recognized the value of them. So.
Yes. Okay. So, Prof. Tan,
Maybe-
Dr. Tan,
yes.
anything last words? Yeah. Melissa?
No, everything is good.
Tan, anything to add?
Yeah,
Or Dr.
everything is
Sabrina?
good.
Any housekeeping? Do we have the e-certificate link up in the chat?
Yes, yes.
Okay, so for the MMA point, please scan the QR code. For the MA point, CPD point, you just use the e-certificate to claim via your via online, I think. Okay, is that correct, Dr. Melissa?
Yeah, yes, that's correct. Yeah.
Yeah.
Thank you.
Okay. Thank you so much, everybody. It has been a very, very pleasurable 2 hours, few minutes session over on a Sunday evening. So enjoy the rest of the day. Please keep safe. Please follow the standard operating procedure for during this pandemic. Hopefully, we will see each other again in the next session. So with that, thank you so much from me.